Hi, everybody. So I think we'll get started.
Yes.
So today I'm going to talk about the tutorial of iCn3D structure viewer. It's a web-based structure viewer. And there are several platforms you can use it. The first one is you can use in a web browser. And, oh, sorry. So if you go to Slack channel and click the channel called presentation, I have these PowerPoint slides in that channel, and you can download it. And because there are many links, you can easily click on the links. So I'll wait for a few minutes, before I really get started, for 1 minute.
Oh, excuse me. Which channel is that in?
Presentation. There's a channel called presentation.
I failed to find that channel.
The Slack channel, and download the slides, because what I want is, at least for the first part, you can follow me step by step and try. You will have some hand-on experience. If you have second computer, that will be even better. So you can use one computer for the presentation, the other one you can try it on.
I'll paste a link to the channel in chat.
Thank you.
So the first part is going to be very basic stuff, and we'll go through step by step. And the second part will be more advanced features. You can just listen, and but you have all the links. If you click the links, you will show exactly what you saw in the slide. So you can come back, do it later. Yeah. Okay, I get started. So my name is Jiyao Wang. I'm a staff scientist at NCBI. Today I'm going to talk about the web-based 3D viewer called iCn3D. So it can be used in different platforms. In web, typically you can open iCn3D in web browser, use this link, or you can Google iCn3D. And you can use it in the Jupyter Notebook. There's instruction. Today I'm going to mainly talk about web use, and the rest you can follow the links and get some hints about how to use it. And you can use Python script or Node.js script to retrieve data from iCn3D. So you can do it for batch analysis for many structures. You can also view iCn3D in a VR headset, for example, Meta Quest 2, or other VR headsets. And you can use Android phone to view iCn3D structures in your surroundings. And there's one option in iCn3D, you can generate models for 3D print. So we have a variety of platforms you can try iCn3D. So first, if you click this link, or Google iCn3D and click the first link, and you will see a UI with several menus. And the first menu is File. So the first thing you want to load is 3D structures. There are two ways to load 3D structures. One way is you can go to Retrieve by ID, and you can try the MNDB or AlphaFold IDs. Here, you can, MNDB ID is the same as PDB ID. I mean, you can try PDB ID or AlphaFold IDs. Basically, it's a uniprobe ID, if you are familiar with protein in uniprobe. And you can input many IDs and show them together, or just input one ID. And the second way is to input your custom PDB files. You can go to Open File, and you can select multiple PDB files or just select one PDB file. And then you can load the structures into iCn3D. And once you load the structure, you can rotate or transform or translate or zoom. So if you use a mouse, you can use left mouse for rotation and the middle mouse for zoom and right mouse for translation. If you're using a Mac or phone, then and click and drag and pinch and spread or two-finger click and drag. Or another way is you can use ladder for rotation, like around around J and K. Around it's L, J, I, N. So around K, there are 4 letters, so you can do different orientation, rotate in different orientations. So once you know how to manipulate the transform the 3D structures, then you can have 3D view and a 2D diagram and a 1D sequence of the sets. So this is a basic slides. If you click the link, you will see exactly the same presentation as I shown here. So in the analysis menu, you can click the first one, view sequence and annotations. This opens up a sequence and annotation window. The top part are the annotation, different kinds of annotations, and the bottom parts are the sequence. And if you click view 2D diagram, it will show, let me try to, okay, I just use mouse. And you will see the diagram for each chain, because the PDB structure typically will have multiple chains, and each chain are colored differently. So here each node is a chain, and you can click one, and then that chain will be selected in your web browser. And if you click the analysis defined sets, you will see all the defined sets. Is my volume okay? Let me make sure that, okay, looks good. So, and when you open the defined sets, so this is the PDB code is 1TUP, and 1TUP underscore A is a chain A, which is brown, and chain B is colored in green. So here you can click each chain and highlight in the 3D structure. So I want to mention, by default, all items are selected, but once you click a chain, you select a chain, and any operation afterward will be focused only on that chain. So I have a toggle near the help menu. So by default, it's all selected. Oops, sorry. So once you select a chain, for example, from the defined sets or from 2D diagram, or you drag on the sequence, and then you select a subset, and this will toggle to selection, and the style and the color will also toggle to orange, indicate that your style and the color will only apply on your selection. On 3D, you can also select directly on 3D. You can press Alt key and click on any residue. And by default, it's selection on residue, but you can select on atom or on each chain. If you click chain, then you click on anywhere near the chain, the whole chain will be selected. Are you following okay? And can you, did you try on your browser? If I'm going too fast, just let me know, I will slow down. Okay, so for example, we select a few residues from the sequence and annotation windows, and then you can click View Selection. Then only that your selection will be shown in the browser. And if you want to go back to the whole structure, you can click here, click the toggle, then it will go back to the full structure. So that's a way to switch back and forth between the whole structure and your selection. As you see, once you select a subset, all the rest sequence will be hidden, only your selection will be shown. And this happens in the same in 2D. And in the sets, your selection will be selected. So it's a synchronized view in 3D, 2D, and 1D. Now I want to show you how to apply different style and color. You have your selection, so you can go to style. In this case, I select schematic. Basically, it's going to be the C alpha trace with the residue labeled with one letter residue name. So it's a neat way to see both the structure and the sequence. And your style will only apply to your selection. And you can show the sidechain nucleotide. And there's also actually nucleotide base as well. This is actually old slide. And you can hide or show the water, and you can show surface here. And electron density map you can show here. And if you want to change the background, you can change to white or black, also in the style menu. I saw some, okay. So now let's talk about how to change color. Again, it's in the color menu. There are many ways to change the color. Unicolor, you can just apply the same color for all residues. Here I choose secondary and choose the secondary structure has sheets and helix. And I choose to show sheets in green. And so that's the secondary structure beta sheets. And you can also color by rainbow or by charge, by atom. And if you click the link, this is share link, it has all the commands to generate this view. So at any point, if you want to view, go to the same view, you can just click the share link. Again, color is applied only on your selection. That's a concept I want to stress. So once you get your custom view, you can save your work. There are two ways to save your work. And I would recommend that you save file, go to iCn3D PNG image, because it will not only save your PNG image, and the PNG image also contains all the commands. And this PNG image can be loaded back to iCn3D. So that's actually the best way to save your work. And you can also, you can even generate a kind of a gallery by concatenating all the HTML source code. So that's another advantage of saving iCn3D PNG image. You can also, this is option 1. Let's talk about another way, which is share link. So option 2, you can share link. This will work if your input is PDB ID or alpha folder ID. If your input is custom PDB file, this will not work. But the previous iCn3D PNG image will work. So in this case, I input the 1TUP, as that's a PDB ID, and the top part is the concatenation of all the commands in a single URL. And there's a limit, it's 4,000 characters. And there's a short link. This is a lifelong short link. You can, send to your colleagues or students, and then they can, just as I show in these slides, I just use this link. So that's a convenient way to communicate. Okay, I think so far, if you have some, hand-on experience, that's pretty much all the basic stuff. Now I'm going to cover more advanced one. You can just listen to it. If you're interested, you can click the links. But otherwise, it's introducing a little more, advanced features. So first, I want to introduce annotations. Annotations are shown in the sequence annotation window, the top part. So there are snips, CLINVA, and conserved domains, and functional sites, and post-translational modification. This is from Uniprop, or interactions. And you can also add your custom annotations. Or you can click to select to show all the annotations, like here. So when you show all the annotations, this is a summary view. So everything is kind of, just show in certain length. And if you click details, then it will show the sequence. So here, top part shows the protein sequence, then follows the conserved domain, CLINVA, SNPs, and these are the functional sites, the binding sites. These are the post-translational modifications and interactions with other chains in the structure. So let's go to next slide. So once you have these annotations, sometimes you want to save them, and you can click this one that's called Save HTML. Then you will save this as an HTML file, basically the same display. But if you really want to retrieve the like residue number, the details, then you can use Node.js script or Python script. And there's a example script called annotation.js at iCn3D GitHub page. So for example, the command to retrieve PTM annotation, post-translational modification annotation, you can just set up a node and type the command node annotation s.js. And this is alpha for Uniprop ID, and the 9 indicated PTM annotations. I assigned 1 digit for each annotations because it's a little overwhelming to retrieve all annotations at once. Each annotation requires backend access. So yeah, it's gonna be a lot if you want to retrieve everything, but sometimes you may be just interested in one of them. If you're interested in everything, then you need to loop through this 1 to 9, and you can still do it. You can also try Python script. One example is at iCn3D GitHub page under iCn3D Python. And this is example script to retrieve the, programmatically retrieve the HTML file. But you can also use Python script to scrape the web page to get all the whatever information you are interested in. So the command for Python script is Python 3, and this, and basically, this script. In this script, you define the URL to retrieve the content. So there's in iCn3D, if you check this script, you will see that a single URL, just like the share link, you will retrieve the HTML source code. So this is more advanced. You can do get annotations programmatically in your command line. And next, I'm going to talk about how to do interaction analysis. Let's say you load a structure, and in this case, it's 3G VU, and there's a GliVec bind to the interact with the protein. Once you load the 3D structure, you can go to analysis, and there's a menu called interaction, and this will pop up. So there are different kinds of interactions. Here I removed the contacts because there are so many contacts. I show the hydrogen bonds, salt bridge, which is in cyan, and pi cation. Pi cation and pi stacking are important interactions in SARS-CoV-2 and ACE2 interaction, also in DNA and RNA interactions. These two, you might not see them very often, but they are important interactions. And then you can select two sets. In this case, I define the ligand, which is this one, and the protein is the rest. And there are different ways to show the interaction. I choose to show to the interaction network. Basically, there are two lines, and one line for the ligand, and the other line for the protein. So it shows interaction between the ligand and the surrounding residues. And the blue is pi stacking, red is pi cation, and the green is hydrogen bonds. So there are strong interactions. You can also see from the dashed lines. And here I label the interacting residues as well. So you can click this to show, click the share link to show this view. And there are other ways you can show in 2D interaction map. Basically, that's X and Y, just like contact map. And there is an even force directed graph that you can try if you are interested in. Okay, I'm going to go to next one. It's a mutation analysis. So if you go to analysis and click mutation, you will see this pop-up window. And you can directly specify, this is PDB ID, the chain, the residue number, and the mutant. And this could be work for AlphaFold as well. And typically you will want to show them in a new page because you don't need to load anything, just in empty iCn3D launch page, and click new page, and you click interactions, this will show up. But if you loaded a structure, for example, your custom PDB file, and then you can specify your mutation and click the current page, then the mutation analysis will show in the current page for your loaded structure. So in this case, it's asparagine at 501 position mutated to tyrosine in the RBD domain of SARS-CoV-2. And it shows actually all the interactions and the common interactions, then also the different interactions in the same window. Here, I just want to focus on the different interactions. So in the Y type, there are 2 hydrogen bonds and a few contacts. Once you mutate to tyrosine, you lost 1 hydrogen, but you gain 1 pi cation and pi stacking interactions. And if you click the link, you will see this view. So some of you might, use this tool, this feature. If you have any questions, just let me know. And iCn3D can also show electrostatic potential for protein, nucleotides, and ligands. So you can show the potential either on surface or as equipotential map. So this might be new to you, but when you show the potential on the surface, the maximum, in this case, the maximum potential is 75 millivolt. Anything above it, it will treat it the same as red. But in your equipotential map, if the potential is higher, then the map will be further away from the, in this case, the ligand. The ligand is showing the equipotential map. So that's a little more accurate, but people usually show potential on the surface. And if you're interested, you can also download the PQR files, which is similar to PDB file, but including the partial charge and the size for each atom. Okay, oops, I think I clicked something wrong. Hold on, I clicked the link somehow. Let me go back to, okay. Hold on. Oops, I lost. Hold on. Somehow I cannot come back to. Okay, let me stop sharing and somehow I cannot see my PowerPoint. Okay, I can see now. Hold on 1 second. Let me share it again. Okay. So I guess you can see my new, okay. Now let's talk about find the similar structures using, so in iCn3D, you can search similar structures. There are different ways to search similar structures. If you want to, for example, find the similar structure for the whole structure, the assembly, then you can go to NCBI VAST and input your PDB ID. And it will show up the VAST display page with similar structures. If you expand each structure, you will see the complex, each assembly, and click iCn3D, you will see the alignment. So that's one way to find the similar structures. And you can also find the similar chains using VAST. So here, instead of input a PDB ID, you input both PDB ID and the chain name. For example, chain A in Wang HHO. And you can click VAST, and it will go to this page. And you can select a few chains and view in iCn3D. And of course, you can also load a PDB file into VAST neighbor search. You will get something similar to this and select multiple chains shown in iCn3D. So once you, from the previous page, if you click the view in iCn3D, you will see the structure aligned, and you will see the sequence aligned based on the structure. So this is the multiple sequence alignment based on the structure alignment. And if you want, you can also directly launch this alignment page by going to File, Align Multiple Chain by Structure Alignment. You can do sequence alignment or residual mapping, but here I choose by structure alignment. You can just input your AlphaFold UniProp ID or the chain name and align with TM align, and you will see something similar like this. So this will be useful if you are interested in certain protein and you want to find which proteins they have the similar structures. And sometimes for example, you have your custom PDB files, and you can load your PDB files, and you cannot do the align multiple chain as I showed in the previous slide, but here you can load multiple PDB files, and then once you got PDB file loaded, you can go to File Realign Selection by structure alignment. The same, you can also by sequence alignment or by residue mapping. But if you do structure alignment, then, for example, you have, in this case, we have 2 AlphaFold structures and 1 PDB structure. And what's underneath is it compares each 2 structures. And once an alignment was found, and then it stops searching because each structure might have multiple chain, right? So like this AlphaFold structure find it's aligned to this chain, then it stopped. It will show this chain. It will not align to the rest chains. And then this structure will also align to the other structure. So this is a way to align multiple structures, but this is also a way to align multiple structure together. For example, if you have one one RBD domain, and if you have 2 different antibody, and then you can load all 3 structures in iCn3D and do the realignment, then the RBD domain will be aligned to one of the antibody, and a different region of the RBD might be aligned to the other antibody. So this is a way you can actually form a complex of RBD with several antibodies. So I think that's pretty much it. And maybe before questions, maybe Jack, you want to show what do you have?
Sure. Give me 1 second. Okay, can everyone see my screen?
Yep.
Okay, cool. So hi, my name is Jack, and I'm just a recent graduate from University of Washington, Tacoma, and I've been working a few months on iCn3D, just some small aspects in it. And one of the things I've been currently working on is creating a legend for all the different colors for iCn3D. So on my screen, you see a web version of iCn3D, and I've loaded in a PDB ID of 1JT0. So when you go under the color tab, you can, so right now currently functioning is the residue and the atom. So if I pull up the legend, it'll tell you to please select an residue or the color from the color menu. And if I select atom, the legend would update, and then it'll show the color or represent the colors that are shown in this 3D model. Similarly, if I change into residue and click on defaults, the legend would update and then show you the corresponding colors for each residue. And that's what I've been working on, I guess, for the past 2 months.
Great.
It's amazing how you take all that work and it's like, oh, it's one feature. But part of why we're doing it, and a couple reasons is one, figuring out how to do things within the iCn3D code base, and the other is thinking about how we use iCn3D in education. So when people are looking at structures, there's a lot of information that's being conveyed, from a chemistry perspective, and the being able to kind of get at it through these legends, we think can help in the classroom. And it's one of the things we put into molecule world is like, okay, so what are the amino acids that are in this protein, and what's where are they when we get into like antibodies and those paratopes. It's really nice to be able to highlight a region and then see the names of the amino acids rather than trying to mouse over them. And so there's an overall effort in how do we start to take this program and adapt it in ways that we can use it in the classroom. I don't know, Sandy, if you have any other.
I think you said it pretty well, Todd. Also,
Okay.
I think what's nice about having the legends is if we look at proteins that have like repetitive amino acid sequences, it looks kind of cool, and you can see, you can quickly identify what the repeats are. So there's a lot of information you can get out of it.
Mm-hmm. Yeah.
Thanks, Jack. So I guess now we should ask, does anybody have questions for Jiyao? Keith?
Yeah.
I have a question. Okay, so iCn3D looks extremely powerful. I would see this being like a whole semester or a quarter for a learning in a class. Give me a little history about it. Is this something that's, and I've never seen it before. So is this something that's used in industry, and how long has it been around? What's it programmed in? Is the programming accessible to users? Little bit more background.
Okay, so about 20 years ago at NCBI, we have a program called iCn3D, which is a standalone application. And about 6 years ago, at that time, WebGL is pretty powerful. So we start to do something similar to iCn3D, but it's a web-based version. Although the code base are different, but many features are similar. So it's JavaScript based, and it's open source in GitHub, and it's freely available to everybody, except there's one license about the Delphi potential, which we use licensed software in the backend. Otherwise, everything else is freely available to everybody. Yeah. So it's been we released in 2016, and it has been there for like 6 years. Over the years, we add more features.
So if I see a protein structure published in an article, would I be safe to assume it's created by iCn3D?
Yeah, I mean, some people use iCn3D engine to generate image, and traditionally people use Pymol or Chimera to generate image. I would say the image quality generated from web-based viewers are not as polished as Pymol, because the ray tracing feature in the web-based version are not as good as the standalone applications. But the image quality should be good enough for publication. Yeah.
Very good. Thank you. It looks extremely powerful. I'd love to dive into it and learn all the features and functions of it. I would imagine it's going to be something that's going to be a necessity if I am pursuing synthetic biology.
It will be. And if I can add to, what makes iCn3D really powerful for teaching in particular is it's web-based, right? So what you don't face is issues with installing software and Pymol, even though you can deal with it if you can deal with the open source version. You can get it open source, but it has licenses. So iCn3D is accessible from a cost and access perspective. So
I want to add that you can also embed iCn3D in your own web page.
Yeah.
You can either just use the iframe version, or you can integrate with your own system. Yeah. There's
even one guy who's using it in a textbook, in a like Open Libre textbook. Henry Chuck. I can't pronounce his last name.
I can remember it. Yeah.
Yeah.
Okay, Angela.
I had a couple of questions and I wanted to, if we could just take a step back, if everyone is okay with that, and if you're okay with it.
Sure.
In the beginner section, you had on your slides for P53, the select on 3D, and you could select by chain, 3D domain, strand helix, residue atom, none.
Yeah.
And I was wondering, I can show it on screen if you want.
Sure. Go ahead.
But I wanted to ask if maybe you could, so you or somebody, if you could explain exactly which one is the chain.
Okay.
And compared to, yeah, which one is, because I saw that you were able to select one item, but somehow I was able to select many things. So how are these defined?
So once you open iCn3D with one TUP, and let's say you open the sequence, and even though this is a basic window, but I want to start with a sequence. And if you show the details, and I want to introduce all annotations.
Okay, here's the sequence right here, and I have them all selected.
So, for example, if you want to click the CleanVar, and these are all the residues which have the CleanVar annotations. If you click on DNA binding site, and these are the residues interacting with the DNA in this chain. So if you scroll, let me hide the rest. So these are just the chain, the sequence alone. And you have multiple, these are the chains, chain A, B, C. And this chain is for the nucleotide. And so if you click them, So that's one way, but the easiest way might be the defined sets. Here you have, you can select multiple of them or select one of them.
So if I select, how do I select them? Do I click on it from here?
If you click here, it's selected. Can you see it's highlighted?
If I click this one.
Yeah, it's selected already.
Oh, hang on a second. I have to go back.
So
here. Like how this one is brown.
Yeah, so if you click on the chain A, it's brown.
Yeah.
So I didn't show you how to select in 3D. So you need to use Alt key. So for example, I think it's Option key in the MacBook, and you click on the residues. So right now it's selecting, oops.
It's selected on the entire.
No, it's selecting one residue. Hold on.
Yeah, of A. Is that right? It's in all of A?
It's selecting on one residue. Let me see. Somehow there's some problem. Let me try
Unless
it.
I can stop sharing my screen and you can share your screen.
Actually, I'm sharing, so you can go ahead. Yeah.
Okay, I'm gonna stop sharing.
Yeah, you can. Okay.
Or do you still want me to share my screen?
Yeah, go ahead. Go ahead.
Okay, I mean, if I'm not sure if this is what people want to use the time, so please interrupt if. Okay, all right. Okay, I'm sharing my screen now.
Yeah.
So I do not have a Mac, I have a PC.
Mm-hmm.
And if I were to, I guess I'm using my mouse and I'm trying to see what I am clicking exactly or selecting.
So you need to click the Alt key and then select on 3D. Press the Alt key.
Press the Alt key. Okay,
Yeah.
and then select on 3D.
Right now, because you select chain, so whenever you click, it select on chain, but you can select residue if you change to residue.
Okay.
Now, if you click Alt key, and it will just select a residue.
Okay, I'm clicking on the Alt key. I mean, I see it's highlighted the
Yeah.
arginine. Maybe it's arginine 202, that's slightly yellow.
Yes, yes.
Okay. Okay, so that's how a residue is selected and versus the chain. Thank you. That was my question.
Yeah.
Yeah, and I'm not sure, Sandra, there was something that we had gone over in our group. Is that there was the loading data issue? Is that something worth
Oh
mentioning?
yeah. I think it's hanging if I do a blast search, Jiyao, but I think I'll just put that into GitHub.
Mm-hmm.
I tried to align a sequence to the sequence from a structure, and blast, well, it's still waiting for blast. So I went back and I clicked Smith Waterman and it worked just fine. So I think it's just a blast thing.
Oh, okay.
But I'll put that
Yeah.
in GitHub.
Okay. Thanks.
Other questions? I think everybody's blown away. All right. Well, I have to say, every time I open CN3D, there's something new. And it's always something new to learn, which is, you know, a good thing and sometimes a challenging thing. But
I want to mention one thing. For the VR view in iCn3D, the Meta Quest 2, it's crystal clear, and it's really worth the $300, and you will have very good view. But otherwise, if you have iPhone, and I just bought this is called Google Daydream View, Oops. So basically, you use the Angular phone, open iCn3D, and then you click the Start VR, and you can put into this. This is like $20. The Google Cardboard is about $7. So these are very cheap VR set, but the quality is not as good as the Meta Quest 2. Yeah.
all right. Thanks, Jiyao.
Okay.
And Jiyao will be with us through the rest of the week too. So be sure if you have questions, you can put them into the, I guess, would you say the help desk section in Slack would be best?
Yeah.
What do you think? Okay. All right. So he'll be able to help us out a little bit more through the rest of the week, which we're really grateful for. Okay. So with that, it's time to work with your teams. You can take breaks. Your team can figure out when you want to take a break. What you need to do for today is to get some kind of presentation ready for tomorrow, so we know what you're going to do the rest of the week, and we can figure out if there's any questions, or if there's anything we want to ask questions about. And I think that's it. So with that, I'm going to leave and I'm going to stop the recording.